welcome to today's webinar for NeuroScientific Biopharmaceuticals. My name is Nicola Gazzatti of Investor Relations Consultancy Corporate Storytime. Today I'm joined by NeuroScientific CEO Stephen Carter, Chairman Paul Rennie and Non-Executive Director Linda Friedland. Paul, I'll now hand over to you to provide the introductions for Stephen and Linda, who have both recently joined the NeuroScientific team.
Yes, thanks very much, Nicola. And good morning and good afternoon to all of the NeuroScientific investors who kindly joined the webinar. The webinar is designed today for our new CEO, Stephen Carter, to provide a deep dive into the M10B technology. But as Nicola said, we have also Linda Friedland on the call. Linda is a physician and also has very extensive commercial experience. So I think for all investors, Linda has joined the NSB board, bring a wealth of experience, and I would just like to pass over to Linda to give us a brief background of your history in the commercial and clinical settings, Linda, and then also I'll go back across to Steve for his presentation. So thank you.
Thanks, Paul. Thank you for that introduction, and good morning to all of you, good afternoon, wherever you may find yourselves. Literally 2 minutes from me. I'm honored and privileged to have joined the NSB board. A minute on my background. As Paul mentioned, my background is in clinical medicine. I trained as a physician, had 25 years in clinical medicine, initially in South Africa. I've been in Australia 15 years, and overlapping that with a long career in the healthcare space, in the pharma space, in the biotech space. I have worked mainly in advisory and corporate consulting and business development for a number of healthcare companies, all the way through from preclinicals through to clinicals, through to full registration, past phase 3 and FDA approvals. My current roles include, for those of you within Western Australia, I'm the Deputy Chairperson for the Neurology Council of Western Australia. I sit on a number of not-for-profit boards. I sit on aged care boards, but primarily my work is in advisory for some Fortune 500 biotech companies. Currently working with some targeted immunotherapy drug development, some brain cancer drug development, some rare diseases in the genetic conditions, and in the neuro space. The reason for saying yes to joining this board is twofold. Firstly, the science and the technology and the drug development I believe in. I've known about the company for many years, but I think in recent times the pivot and the change in management instills a lot of confidence in me. I think there's potential, and I have confidence in the future success of NeuroScientific. The second reason is the people. You can have really superb compounds, and without the right people, no success. And I have known Paul for not that long, over a year or so, but I know of his success. I know of his leadership style, and I am delighted to work with him. I've known Stephen Quantrill for many years, and I've known Abby for a period of time, and it is truly a superb board. Anton, I've recently met, superb board. I've recently met Steve Carter when he came on board, and I have full confidence in the management team and the scientists, and I'm really happy to be on this board and hope to add value. So that's all from me. Looking forward to good success. I hand over to our CEO, who's going to give you a real deep dive into the company at the moment.
As mentioned, my name's Steve Carter. I've actually been involved in the biotech industry and the pharmaceutical industry for over 30 years. And I started on the bench. So I'm a scientist by background. I'm a chemist, biochemist. And I started as a research and developer, developing a number of anti-cancer drugs for companies in Perth, and then worked with Upjohn. I was a research and development manager for Upjohn. And then when Upjohn and Pharmacia came together, I actually was moved up to run the whole of the manufacturing facility. We were manufacturing millions of units a day. We had a lot of products, sterile, from creams through to sterile anti-cancer drugs, and then moved into the general management of the company. From then, for the last roughly 20 years, I've been running biotechnology companies, developing drugs from scratch, basically, including one which we had to grow the plants to extract the drug from, make the original drug, and then take that through into the clinic. So I've developed a lot of drugs for many different therapeutic indications and run them through the FDA, the EMA, so the Europeans, the Americans, the Australians, done a lot of clinical trials in Australia, the US, and in Africa. So have a very good understanding of the drug development process and what's required. When I was started looking at NSB, I liked what I saw. It really did excite me. And the more I've looked at it, the more I like it. And I think, again, as Linda said, we've got a great board, we've got a great team here, and I look forward to moving forward with this company. And I hope the shareholders enjoy the presentation and agree with the board's decision to move into the ophthalmologic area. Thank you.
Thank you so much to you both for your introduction and giving us a bit of your backgrounds. We'll now proceed with the presentation from Stephen, who will be providing us with an update following the company's announced September quarterly activities report, which outlines the intention to advance lead drug compound EMTB as a therapy for neurodegenerative conditions of the eye, such as glaucoma. We will then follow this by a Q&A session. And if you do have any questions, please use the Q&A function at the bottom of your screen, and I will put your question to Stephen at the conclusion of his presentation. So without any further ado, I'm pleased to hand over to Stephen.
Okay, well, thank you for that, and welcome to the NeuroScientific Investor Update webinar. This is my first with the company, and I'm very pleased to be able to discuss our strategy with you today. We have just our standard disclaimer. It explains that we will be making a number of forward-looking statements in this presentation, and this should not be relied on as a recommendation to buy or sell NSB stock. For those that have only just coming on board, a little bit of background on the product. NSB are developing EMTB for the treatment of neurodegenerative diseases. EMTB is a four-armed or a tetrameric dendrimer peptide, and it's been modelled on the beta domain of metallothionein. Metallothionein is a naturally occurring protein within the body that's very important in a number of different areas. Tissue protection, regulation of metal ions, it protects against oxidative stress. But the one we're really interested in is a neuroprotection and neuroregeneration. When we look at the metallothionein molecule, there's a number of areas. There's a domain alpha and a domain beta that are involved in this neuroprotection, regeneration, memory aspect. We've optimized our EMTB to focus on the beta domain, which is about neuroprotection and regeneration. So that's the focus, and the name EMTB is EMTM to do with the beta. We have an EMTMA as well. So since I started with the company in mid-September, I've spent the whole time working with the board and the executive team and our key consultants, conducting a full analysis and review of the NeuroScientific research and development programs. From the results of this review, confirmed clearly in my mind that NeuroScientific's board, management, and the shareholders have very good reasons to be positive about EMTB's therapeutic potential. The early non-clinical and preclinical studies carried out across a wide range of animal studies confirm that EMTB is an exciting lead prospect with a significant therapeutic potential. And if we are able to successfully develop this project and show it's safe to use, it could be a game-changing product in the fight against neurodegenerative diseases. Just a few of the project highlights, and I'll go into these in a little bit more detail. We know the cellular target. It's a molecule called LRP1. We know the mechanism of action, and we know this opens up multiple therapeutic applications. Our studies to date have confirmed that EMTB is neuroprotective, it's neuroregenerative. It regenerates nerves, it regenerates the myelin protective coat that surrounds the nerves. We know it can slow cognitive decline in animal models. We know we can actually slow optic nerve damage, and it's also neuroprotective to the optic nerve. We also understand that it is absorbed into all the major optic tissues, and all the work we've done to date shows this product is actually safe. So what we have here, I'll just go through a little bit about how the M10B works on the cell. So we have a nerve cell, and on top of the nerve cell, we have this LRP1, or a low-density lipoprotein receptor-related protein. These are quite specific in what they do, as I mentioned. We bind to that to the binding sites, the lipid binding sites on LRP1, and when we bind, we trigger a intracellular signaling pathway, which signals for the cell to manufacture a number of activation proteins. The ERK, AKT, CREB are a few of those. And when we activate those, we see that they will protect the neurons. So we have this neuroprotective effect. They also downregulate the neuroinflammation, and they modulate differentiation, allows the cell to start to repair itself. What I'm going to show you now is just a short video on how this actually works, and maybe a more visual aspect of how M10B actually works. So we have neurons that are nerve cells, and they receive and transmit the information within the central nervous system. So we're seeing the signal go down through the nerves. When the nerve gets damaged, the body doesn't normally replace that nerve, and so you break the cycle. We put in M10B, which is this the X coming onto the screen. It binds to the LRP1 receptor, which is on top of the nerve cell. That generates these signaling pathways, which will then allow the nerve to start to regenerate itself, and then the signal can pass on the way down. So how do we know that we're actually binding to LRP1? How do we know that's our target? Well, we've done a number of experiments, and the figure 3 that we can see there is an experiment which we did, where we put different concentrations of M10B into a solution of these LRPs. What we see over the first 10 minutes, that first 600 seconds, we see an increase in concentration of the M10B as it solubilizes. And then at around about the 10-minute mark, we actually see the concentration dropping off. That's an indication that the M10B is now being bound up by the LRP1 receptor. But is it specific to the LRP1 receptor, or is it or not? So we did another experiment, which is what the one showing in figure 4, using another protein as well as LRP1, a protein called a receptor-associated protein, which is actually a molecular chaperone. It's known to bind very strongly to the LRP processes. So again, we put the M10B in, we see the increase of concentration, but now at 10 minutes, it still continues. We actually don't see it binding to the LRP. If we look at the RAP protein, the bottom line, we see at around about 20 minutes, the concentration of that increases. So the M10B is actually displacing the RAP from the LRP. The M10B concentration goes down, the RAP concentration goes up. So we're seeing, yes, it's very specific to the LRP1. So we know what we're targeting on the cells. How do we know, though, that when we target that, are we seeing those signaling pathways? Yes, we do. And what we've got here is, again, we've used M10B, and we've put it in and then measured whether the various signaling pathways increase. And what we see across the board here, whether it's ERK, AKT, or CREB, we see an increase, up to a 50% increase. And we see also concentration-related increases, especially with AKT. So we are seeing that, 1, we bind to the LRP, and 2, we do produce these special neuroprotective proteins that we're interested in. And we've seen that we do that. Does it actually make a difference when we get into the cell? Again, the answer is yes. So that first graph shows that we are stimulating survival of neurons. We have over greater than 90% improvement in survival of the neurons during the experiment. We also promote regeneration. So if you look at the cortical and the hippocampal from the brain, and we look at the spinal cord neurons, in all of them, we see a massive increase in the length of those neurons. So those neurons are growing, where we see nearly 400% increase growing from those damaged neurons to the new ones. Okay, we grow neurons, but are we generating new neurons, and are we improving the ability for them to work? Well, we have, when you have two nerves come together, between two nerves is a synapse, which is a structure which permits the neuron or the nerve cell to pass an electrical signal between one and the other, and that's how nerves work. So if we were to see an increase in the number of synapses, it means we're getting an increase in the number of nerves that are being generated. And what do we see in that last one? We see up to a 10-fold increase in the synaptic density. So yes, we are. We are definitely protecting, regenerating, and getting new nerves. So the information provides a very strong argument for the drug's ability to specifically target the neuroprotective and regenerative targeting signaling pathways. If we look at the safety of the drug, we've done a lot of studies around that. We've looked at the central nervous system safety, the cardiovascular safety, the respiratory safety, and we've confirmed in all of those studies that there is no safety concerns using M10B. We've also shown that it doesn't damage cells and mutate those cells, and it's not toxic to the DNA or the other genetic material in there, which is very important with the drugs. We've looked at the macroscope. We've looked at the organs. We've looked at whether the dogs and cats and monkeys lose weight, whether they go off their food, whether there's any changes to the heart rhythm. All of these, we saw nothing like that occurring. So again, M10B is safe to use. We did see some irritation around the injection sites when we were doing some of the injection work. We identified that as being an issue with the formulation. We reformulated the drug, and the irritation around those injection sites was significantly reduced. We did have a payoff with the formulation being a little bit less stable. Moving. Oh, data supports safety. Sorry. Okay, we look at let's look at the systemic projects. These are the multiple sclerosis and the Alzheimer's disease work that was done. And we did show considerable therapeutic potential in both of those areas. In our preclinical model for multiple sclerosis, we saw a significant difference in the functional improvement of the M10B treated animals. We've also evaluated M10B with another multiple sclerosis drug, Copaxone, and we saw there that putting those two drugs together, we actually saw an additive effect. So 1 and 1 didn't equal 2, 1 and 1 equals 3. So definitely a significant benefit there, and those systemic models confirmed that if we were able to get the drug to the target sites, then there was the potential to see functional improvement. Just want to look at the eye and to see what occurs at the eye. So this is a quick video just to sort of show you around the eye and a little bit about glaucoma, so it gives a bit of background up to what we're looking at. So the optic nerve transmits visual information from the eye to the brain. So when we expand that, we see within the eye, we've got a number of the retina, the optic nerve, the vitreous humor, and the retina. If we want to look more deeply into the retina and just have a look what's occurring in the retina, we see a number of cells which are called retinal ganglion cells, and these are the cells that will transmit the light images down the optic nerve to the brain through the process. If they get damaged, we lose vision, and it can lead to permanent vision loss if we can't fix those. So these retinal ganglion cells, as I said, go right the way back, down, join into the optic nerve and move down. Now, if we just have a quick look at the eye and what happens during glaucoma. During glaucoma, the pressure inside the eye it increases, and that puts pressure on the nerves and damages the nerves. It damages those retinal ganglion cells. It damages the optic nerve to the stage where you get this degeneration occurring. So the nerves are damaged. M10B, as it gets into the eye, again, like we saw earlier on, will bind to the LRP1 receptor. It sends out the signaling pathway and it regenerates the nerve. So we see that this protective effect within the eye, and we also see this regenerative effect within the nerves, regenerating them when we put M10B in there. So once we regenerate these axons, they regrow and reintegrate into the optic nerve and allow the optic nerve to either be protected and to also regenerate and grow. Does that really happen in real life? Well, yes. Here's a study that was done not using M10B, but using the whole protein metallothionein. And we've taken the section of metallothionein, which we know works in neuroregeneration. That's our M10B. So in this study, the top picture, the untreated optic nerves, that to me is very exciting. So we injected M10B into mice and had a look at where it went. And so if we look at the first set of pictures there on the left-hand side, the retinal tissues, The green that you see is the actual MTMB. So after 24 hours, you see a significant amount of MTMB in that retinal tissue, and it's still there after 6 days. When we look at the optic nerve to see whether we get the optic nerves, we need to get there for it to be effective. Again, after 24 hours, we see it in the optic nerve, and again, after 6 days, it's still there. Okay, but we've got it there. It doesn't do anything once we get there. So we wanted to have a look at whether those signaling pathways were initiated. So the far right pictures are showing you that, yes, the untreated eye, you didn't see a lot of generation of those signaling pathways, and the treated eye, you actually did. They're a lot brighter and a lot more of those signaling pathways being opened and used. So we can get it into the eye, we can get it to the tissues, and when it gets to the tissues, it does what we want it to do in getting those signals working. So how do we get it into the eye? Well, we actually inject it into the eye in what's called an in vitro injection. Now, this doesn't sound very nice, but it's actually a very common way of getting drugs into the eye. For example, macular degeneration. If you've got macular degeneration, you'll get a monthly injection into the eye for the rest of your life to try and minimize the damage that's caused. So it is quite common. And my brother-in-law had macular degeneration. He said it was not a problem getting an injection in the eye. He didn't find it a concern at all. So we've looked at this. Okay, if we do inject into the eye like in this manner, do we see it in there? And yes, we've seen it for up to 14 days after an IVT injection. We found it to distribute right throughout the eye. So in the vitreous humor, which is the large orbital section of the eye, in the aqueous humor, which is right at the front, in front of the pupil there, behind the cornea, and also down into the optic nerves. So, and we saw it there for 14 days, which is quite surprising, because when we look at putting it into the systemic system and injecting into the bloodstream, it doesn't last for any more than 30 hours. So having it there for that longer period of time means that we don't have to do very, very many injections. We can do 1 injection a month or 1 injection every 2 months, and that'll come out over the work we do coming up. We did carry out a 13-week study in non-human primates, looking at multiple injections. So not just 1 injection, 4 injections, and just see whether we could see the drug in the eye. And yes, no matter what concentration we put it in, we actually saw the drug in the eye for that period of time. And from that, we actually determined that we had what the safe injection concentration would be for our first in-human studies. We are required to do a second animal study to confirm that data, and that animal study is currently being planned and should be underway soon. We need intellectual property. Intellectual property is really it's what we have, it's what we sell, it's what we're developing. And the proposed route of administration and our therapeutic focus moving into these ophthalmology projects increases our ability to increase our intellectual property. So we are looking forward to putting more intellectual property in. Also, there are a number of other neurological disorders, ocular diseases, such as ischemic optic neuropathy, which have a smaller number of people and allow us to access what's called the orphan drug programs. Orphan drug programs, most major regulatory groups have them. The FDA has one, the EMA has one, the TGA in Australia has one. What they do is they allow you to have exclusive rights in the market for example, in the USA for 7 years after your marketing authorization, in Europe 10 years after your marketing authorization. So it's like having a patent. Nobody can compete against you in that market, in those markets. So we're looking at what orphan drug work we can get underway as well. Now, just to give a little bit of background on glaucoma, because glaucoma is a massive opportunity. It's the leading cause of irreversible blindness in Australia, and it's rapidly increasing its prevalence due to our aging population. The global prevalence of glaucoma in 2020 was 80 million cases, of which 6 million of these people were bilaterally blind because of the end-stage disease. In Australia, the estimated prevalence is about 4%, but that's increasing, and by 2025, it'll be about 80% greater than that. If you're looking at 80-year-olds, people above, it's actually 10%, so it's quite a significant amount. And 50% of the people who have glaucoma don't know, because it's an asymptomatic disease. It's not until you start to see damage occurring to your eyes that it would normally be picked up. So, and that 50%, that is greatly increased when you move into the minority groups, such as the Australian Aboriginals and the socioeconomic people groups as well. New artificial intelligence scanning technology is now coming to being, where they can scan the eye with a simple camera, and they can diagnose different diseases, including glaucoma, macular degeneration, and others. And in fact, in America, they've actually now started rolling this program out in pharmacies, where you walk in, you pay your $20, you put your face up against these cameras, they take a photo, the AI will work out whether you've got an optic disease, and if you have, it'll give you, recommend that you go and see your ophthalmologist and give you some information to take to them. Why is that important? Well, it's going to give us a greater ability to get earlier diagnosis, and earlier diagnosis is very important in these diseases. You go through 3 stages of optical disease. One, the adaptive, where you've got some oxidative stress and some neuronal dysfunction. At that point, if you can get in and protect the neurons and maintain and preserve the current visual function, that's the best time to do it. Then you move into the early pathology, where you have oxidative stress, the neuronal dysfunction and vascular dysfunction, and you're starting to get reversible damage. You're losing eye function, but if you can treat it with M10B here, you can see neuroregeneration and a repairing of the vision potentially there. Once you move into late pathology, the irreversible damage has occurred, and then it becomes very difficult and very complex treatment programs to try and get that right. So the earlier we can diagnose, the earlier we can treat, the better the outcome. Glaucoma is just one of many potential neurodegenerative diseases that are out there. Glaucoma is not done at home, such as with multiple sclerosis and Alzheimer's disease. The treatment protocol was to give daily injections at home. Here, we carry it out at a medical facility by a doctor. So that changes the whole product dynamics. It changes the amount of drug we give. It changes the stability requirements that we need. It changes the formulation requirements, because we've got a much robust treatment background protocol as it's being carried out by medical specialists. So therefore, a stability of a few hours, which wouldn't be acceptable in a home environment, is now acceptable in a medical environment, because the doctor is going to make the drug up and use it while you're there. It's not going to be made up the day before. It's not going to be made up in the morning. It's not something like that. They're going to be making up and using it then. Also, the fact that we've got a low dose and only a once monthly or once every 2 monthly injection, as well as the size of the market, provides a really strong health economic evaluation and a potentially significant margin for the product owner. We have some significant preclinical data that shows there is a strong argument for developing M10B for ocular disease. So this next slide, I know it's a little bit busy, but the idea of this is to show you just the significant amount of work that we've already done, which is relevant to the ocular program that we're going here. We've done the lead optimization. We now have a product which we know works in neurodegeneration, neuroregeneration, and so forth. So that's been a lot of work done for that. That's all specific, will work for any therapeutic use, and especially for the use going forward. We've done some animal efficacy models. So we've looked in rabbits, we've looked in pigs, we've done in vitro injections. We've confirmed that it has a neuroprotective effect. We've also confirmed that it permeates all tissues. We've looked at the safety, and we've done a number of studies, ophthalmologic studies in these, and we've done a number of studies that were carried out for both, with regard to whatever the therapeutic use of the drug is. That's all will be used. We currently have a 28-day study, which is we're waiting for it to be reported on. And as I mentioned earlier on, we've got this 13-week study in rabbits to give us the final information for the phase 1 study. We're putting all that information together, all this information we've got, we're putting it together in a pre-IND type B meeting. So that's a meeting with the FDA that will go through all the data we've got, our plans moving forward, and we ask for their advice and their recommendations. The idea of that is to ensure that as we move forward, we're moving forward in a manner which is going to be acceptable to the key regulatory authorities around the world. So that process is already started, and we hope to, it takes a couple of months to go through the whole process, but at the end of it, we should have a very good document and a very good rapport with the FDA. Further to all the data we've got here, all the work that we've done on multiple sclerosis and Alzheimer's, that's all very supportive for us moving forward, and will be able to be used in certain areas. Commercialization, how do we commercialize the product? Well, we're using a very similar commercialization strategy to most biotechnologies companies. We need to take it through the early clinical development, show the drug is safe, show that we've got some efficacy markers coming up in an early study, and then in phase 2, start talking about licensing and see if we can license the product out, and then work with our partner for the phase 3 studies, the large phase 3 studies. So we're moving into that phase 1 area. So we've actually come about a fair way through. There's a lot of work being done, and now we're virtually ready to move into humans. And once we get these last study or 2 done that I talked about. Do we have a plan to work forward on that? Yes, we do. We have a strategy, and we have an indicative schedule. But I really need to point out that whilst these milestones are what we believe can be achieved and what we're aiming for, and this is what we're measuring ourselves on, these dates are subject to change due to the reliance on independent third-party contract research organisations, and we don't control their timetables. So we need them to undertake and successfully complete each stage in the timeframes that we've agreed. So as we move forward, so this is the document we're going to be working to, but these timeframes may change. Hopefully we can change them, bring them forward, but rather than pushing them out. But as I said, we have independent third-party groups which are involved, and that we don't have control over them. We have a board and management team with a great deal of experience. Linda's given her experience, and that is phenomenal for a company of our size to have someone of Linda's quality coming on board. Paul, he's in phase 3. Paul's in phase 3 studies with Paradigm. He's walked this walk. He knows exactly what he's doing. He understands this. He knows both from a corporate and development point of view what's required, and brings a great deal of strength. Anton has also walked this walk. He's the founding director of a number of biotech companies. He understands what's required, and also, again, has good corporate background. Both Stephen and Abby have got a great sort of investment professionals. They have very strong corporate advisory advice and corporate governance. And myself, I gave you a bit of my background earlier on, 30 years in senior roles in drug development. So I think when you look at the team, the board's put together a good, strong board and a good, strong management team to take this project forward for you. Where are we at financially? We've currently got roughly $4 million on hand. We believe that's sufficient cash to develop our lead product into the clinical trials. We have 143 million shares on issue. They are tightly held, with the top 20 holding 53% of that. Macrae Investments, senior major shareholder, at 18%, and BNP Paribas at 13%. That's mainly one company called Alpha Swiss. So that gives us an idea that we have a strong shareholder base. So in summary, the company has conducted the M10B project review, and we confirm that M10B is our prospective lead project. M10B for the treatment of neurodegenerative disease of the eye has shown great promise. There is a huge market potential, with greater than 5% of the global population suffering vision loss due to damaged optic nerves. So that is a very addressable, accessible market. Ocular disease has outstanding health economic profiles from our point of view. M10B for the development of ocular diseases is definitely commercially viable. So we are going to, as a company, focus on the developing of M10B for the treatment of glaucoma and other neurodegenerative diseases of the eye. And we have the management team and the leadership team expertise to ensure that can occur. So thank you very much. That's me finished.
Thank you, Stephen, for that comprehensive update. We'll now move on to our Q&A session today. Our first question is in 2 parts. The previous management spent a lot of time and energy progressing M10B as a potential therapy for diseases like Alzheimer's and MS. Will this work be useful to your R&D into M10's application for diseases like glaucoma? And the second part of the question, what would management say to shareholders who have purchased and held on the basis of representations made regarding the prospectivity of MS and Alzheimer's applications for M10B in light of the September quarterly activity report?
Thanks, Nicola. They're both very good questions, and I'll answer that first section first. So the simple answer to the first part of that question, with regard to will the work be suitable? The simple answer is yes. As I went through in the presentation, there's been a significant amount of work done that is applicable to both streams. So that work will continue, that data will be continually be used and will be used from both a marketing side and also from a regulatory aspect. So that data is still very usable. Some of the early work that was done was exploratory and can never be used for regulatory processes, but that's how drug development works. You need to use that, you need to do that exploratory work to determine what your therapeutic path is, to determine your therapeutic progress, to determine the doses you require. And that work is carried out to allow management and to make the decisions, such as the decision we've made, is what is the best commercial and therapeutic direction for the company to move forward this project. And that's what we did. That's what we've just done, is looked at all that data. Yes, we can use a lot of it, and we believe the decisions we've made will be for the benefit of the shareholders there. With regard to the second aspect of the question, what would we say to shareholders who purchased and held shares on the basis of representations? I'd say, well, I'm glad you've held the shares. I think you're going to see a significant, improvement over the coming years as we develop this product. I know that some of the shareholders may have bought those shares because they had an attachment, or they had some emotional attachment to both Alzheimer's or multiple sclerosis. I hope those shareholders remain with us, because what we're doing is now we're still working in the neurodegenerative diseases, moving into glaucoma and other optic diseases, which cover a significant part of the population. As we said, greater 5% of the world's population have these. And if we don't treat them, some of those are going to turn blind, and we can actually stop that. It's a great benefit. So yes, a work was done. I'm glad we needed to do that work. We have now looked at what is going to be the most worthwhile process forward, what is going to give the biggest commercial return to the shareholders, and what is our best chance therapeutically to get a drug forward and moved into the optic area.
Thank you, Stephen. Our second question, are you confident that the Human Research Ethics Committee issues, which held up approvals for the phase 1 clinical trials last year, are not likely to be repeated for the clinical trials you are now planning?
Yes, again, a good question, and one I'm sure that most shareholders would like to understand. And yes, I am confident for a number of reasons. One, the majority of the concerns brought up by the HREC, we've overcome those. We've done that work, but we're moving into a different therapeutic focus. We're moving into a different route of administration and so forth. So so that assists some of those things. Would not be an issue, because we're injecting this directly into the eye, where we're not going to see the systemic use of the toxicity of the drug that you may see otherwise. We're also planning to meet with the FDA. We're going to sit down and discuss with the FDA everything that we've got, how it relates to our program going forward, what our plan moving into human trials is, and the FDA will then provide us with the feedback. I prefer you look at this, or we'll look at that. So we will overcome those issues long before we get to the HREC aspect. We understand things such as our mechanism of action. We understand how the drug gets into the target tissues. We know that we can get into these target tissues where we didn't actually know that when we were going in a systemic manner. We hoped they would go in. We've seen it work in animals, but we know we're now injecting directly into the tissue, therefore getting it. So we, again, overcoming some of those potential concerns. And it's going to be, the drug will be administered in a control environment, again, changing the dynamics of the drug development process, therefore changing some of the concerns that a HREC may bring up.
Thank you, Stephen, for your answers to the questions. That does appear to cover the majority of the questions from our audience today. And if you do have any further questions for the NeuroScientific team, please email them to info@corporatestorytime.com. This does conclude our webinar for today, so I thank you all for attending, and thank you, Stephen and the NeuroScientific team, for the comprehensive update.